Well, I think we can potentially get started. Hello, everybody. Welcome to Australasian Mycorrhizal Society's first virtual seminar. Thank you for registering and joining us today. Hopefully the registration process wasn't too difficult. We are learning from this experience, and as it is our first one, hopefully it goes smoothly. Please make sure that your microphones and videos are turned off to enable the best video streaming quality and less disruptions. I'd like to start by acknowledging the traditional owners of the land that we're meeting on today across Australia and New Zealand, and pay my respects to their elders past and present. My name is Tracy Steinricken, and I'm the president of the Australasian Mycorrhizal Society. As this is our first seminar, bear with us if there's any technical issues. We're going to save all the questions to the end of the presentation, as we've allotted 15 minutes for a discussion. You can write out any questions in the chat bar for me to ask them to Stephanie when she's finished talking. Or if you'd like to ask her directly at the end of her talk, select the hands up button on the bottom of your screen. There's a toolbar that says reactions. You can put your hand up there. And you can then ask next using your video and microphone. Getting to today's speaker, Stephanie-Watts Fawkes will be presenting her talk today titled Identifying and Characterising a Zinc Transporter Involved in the Arbuscular Mycorrhizal Symbiosis. Stephanie was an independent Ramsay Fellow at the University of Adelaide's Wake campus. There, she conducted research on the plant-soil fungus symbiosis known as arbuscular mycorrhizas and investigated how they improve plant zinc and phosphorus nutrition on nutrient-depleted soils. She uses a combination of molecular and physiological techniques to study the symbiosis, and she is interested in the potential agricultural applications of arbuscular mycorrhizas, particularly for food security purposes. Her talk today discusses the work she did while at the University of Adelaide. Stephanie, when you're ready, go for it.
Fantastic. Thanks, Tracy, for that introduction. And thanks so much for hosting me today. So the work that I'm presenting to you is part of, as Tracy mentioned, the fellowship that I did over 4 years at University of Adelaide, which I only completed a few months ago. And I would like to give you a bit of a summary of sort of the main topic that I studied as part of the fellowship. So for those of you who aren't familiar with mycorrhizal fungi, or AMF, they can colonize, they live in the soil, and they can colonize the vast majority of our terrestrial plant species. And this includes many important crop species, wheat, barley, oats, and also a lot of horticultural species. So they're quite important for the management of our agriculture. And the main benefit of the symbiosis between mycorrhizal fungi and plants is that they can improve plant growth or yield, and they can also improve the plant's nutritional status. And this is because the symbiosis, when forms here in root cortical cells, you can see in this cartoon here, and these fungal structures called arbuscules are formed. And this actually is a site of nutrient transfer between the plant and the fungus. So mycorrhizal fungi can take up nutrients from the soil and transfer them into the plant via these arbuscules. And in return, the plant can provide the fungus with a source of a fuel source, and able to grow and reproduce and acquire more nutrients. So it really is an association where both partners are getting something out of it. So mycorrhizal fungi can particularly be beneficial when the host plant is under some kind of nutrient stress from the soil. Typically, this would be a deficiency, for example, of phosphorus or zinc or other micronutrients or macronutrients. However, in some cases, the nutrient stress may also be a toxicity. For example, zinc can be present in toxic amounts, and mycorrhizal fungi have been shown to also alleviate nutrient stress that is as a toxic situation. This is just a photo under the microscope that my previous master's student DM took. This is a Medicago truncatula root that's been colonized by an AMF, I think Rhizophagus irregularis, which is usually the species that we use in our glasshouse and studies. And you can see all this blue material here in the cortical root cells, this is all fungal material. There's a lot of arbuscules, which are these sort of the tree-like structures that look kind of like this. And then there are these sort of blue, darker blue bubbles here that are vesicles, which are sort of storage compounds for the fungus. And the rest of the material is intercellular hyphae that runs from cell to cell. So this is a really nicely and well colonized root, just as an example. I'm just going to see if I can move that. There we go. So as I mentioned, soil nutrient stress or alleviation of soil nutrient stress is one of the benefits of being a mycorrhizal plant. And in particular, soil zinc deficiency and the effects of mycorrhizal fungi have been my primary interest for pretty much the last 10 years since I started my honours. And this is because soil zinc deficiency is actually quite prevalent. It's widespread and it's common. Micronutrient deficiency in the soil, and it affects how crops grow. In fact, it's estimated to affect 50% of the world's cereal growing soils. And if you have a look at this map and have a look at the regions of high prevalence of zinc deficiency, you'll notice that these locations tend to also be in developing countries that may rely more heavily on cereals in their diet. So it's really important to me to work out how we can use mycorrhizal fungi to improve plant zinc nutrition. You can also see in Australia there are a few areas that coincide particularly with our southern and our western cropping regions that are also low in soil zinc. So now I want to talk about how plants obtain their zinc. And if you think of a plant growing its roots into the soil, we have two pathways of zinc uptake. We have what we call the direct pathway of uptake, which is via root hairs, which is if you have a non-mycorrhizal plant, this is how they would obtain all their zinc from the soil. The root hairs acquire zinc and transport it into the plant. When a plant associates with mycorrhizal fungi in the soil, this opens up a second pathway of nutrient uptake. And this pink thing here is the fungus, and this is the external hyphae that are reaching out into the soil. Basically, or simplified, it's an extension of the root system. These external hyphae go out and search for nutrients. Zinc is one of them, but it's not limited to zinc. There's also phosphorus and iron and other micronutrients as well. But basically what happens is the hyphae can acquire zinc from the soil and transport that back into the plant via the mycorrhizal pathway, and where they've colonized cortical root cells with these very specialized structures called arbuscules, which if you remember from a couple of slides ago, these arbuscules form the site of nutrient transfer between plant and the fungus. So the fungus is acquiring zinc, and then this gets into the plant via a specialized membrane, which is a plant membrane that basically sits around the arbuscule, and it's called the periarbuscular membrane. So previously, myself and other researchers from the physiological side, we've used radioisotopes of zinc, so radioactive zinc, to label the soil. And we've seen whether or how much zinc of that radioactive zinc gets into the plant, and quantified how much zinc is coming through the mycorrhizal pathway. So that's something that I did a few years ago, and also as part of my PhD. So we've kind of shown as proof of concept that zinc does come through this mycorrhizal pathway of uptake. So what remains unknown is sort of the molecular side of that pathway. If you think about those two pathways again, we have some idea of which genes and which transporters are involved with the direct pathway of uptake. So via this pathway here, we have some idea of what transporters are responsible for that movement of zinc. However, when it comes to the mycorrhizal pathway, we have basically no idea what genes, what transporters are responsible for transporting zinc there. And so in this case, we're looking for something that would be on this periarbuscular membrane where the other transporters have been discovered for other nutrients that are involved in the mycorrhizal pathway of uptake. So this question really drove my independent fellowship, the Ramsay fellowship that I undertook at Adelaide Uni. And I guess the really overarching question was, what are the molecular mechanisms behind mycorrhizal pathway zinc uptake? So in starting this project, having really no basis or other research to build on for this question, I needed to develop some really strong and clear hypotheses for how I was going to undertake answering this question. So I'd just like to go through these hypotheses now, because I think it really helps explain to people why I've done what I've done to answer this question. So the first hypothesis is that the candidate gene, so the gene that's involved for the zinc transport in across the periarbuscular membrane, that this gene is expressed and it's upregulated in the roots of plants that are colonized by mycorrhizal fungi. So this is in comparison to a control plant that's not colonized. So we expect the gene would be more highly expressed. Secondly, and this might seem quite obvious, but it's very important to test this properly, is that the candidate protein is able to transport zinc. So it's one thing to say that it's annotated, the gene's annotated as a zinc transporter, but it's another thing again to put that in a system where you can get strong evidence that it's actually zinc that's being transported across the membrane by this transporter. Thirdly, you know, I've talked about this specialized membrane quite a bit, and I would expect that if the candidate protein has this role, then it should be localized to that periarbuscular membrane. It should be there doing the role of transporting zinc from fungus into plant. And lastly, although this is not essential to finding this gene or finding the molecular mechanisms, this is really interesting part of this work, which is the hypothesis that if you have a plant that doesn't have a functional gene of this candidate gene, that there's some kind of phenotype that you can measure compared to the wild type that does have a functional gene. So this might be slower growth or lower total biomass by the end of an experiment. Given that I expect it to be involved in zinc uptake, I would be looking at total amounts of zinc in the tissues to see whether there's a difference between a plant that has a functional gene compared to one that doesn't have that functional. So now this kind of leads into where I started. So how did I even begin to grapple with finding this gene? So I took, I guess, an educated guess, which is that we already know something about how zinc is acquired by the direct pathway, so by that root hair pathway. And we know that zinc is acquired by transporters that are called zip transporters. So when I started this project, the most obvious direction to take was to look into whether there are zips on the periarbuscular membrane that may be responsible for zinc transport by the mycorrhizal pathway. And at exactly the same time as I started my fellowship in 2016, there was a really neat review published on heavy metal transport in the mycorrhizal symbiosis. And that review also hypothesized that on this plant membrane here, that there is a zip that's responsible for zinc transport. But during my fellowship, there was no other reports or publications that I know of that came out to suggest whether that's true, or what that zip was. So I continued, or I started, I guess, with a transcriptome analysis. So for those of you, I'm not really sure what the background of the attendees are today, but for those of you who aren't familiar with RNA sequencing, basically this involves taking RNA from roots. In this case, I chose the model legume Medicago truncatula to conduct all my studies on, as it's really nice. It has a really nice molecular toolkit for doing this kind of fundamental research, and it also gets colonized by mycorrhizal fungi. So I extracted RNA from Medicago plants that were colonized and that were not colonized by mycorrhizal fungi. And RNA sequencing basically allows you to look at the expression of every gene in that plant all at once. and give you a relative measure of expression of those genes. So it's a neat way to sort of start an exploratory analysis and then hone in on what, a list of candidate genes from that to help get to your final answer. So, so I did that. I grew metacargo plants with and without mycorrhizal colonization. I also grew them at 3 different zinc concentrations because I wanted to be sure that the gene that I selected to work on further showed a upregulation of expression across different zinc treatments and not just at high zinc or particularly at low zinc. And there were about 1,000 genes in total that were upregulated in the mycorrhizal plants. And this Venn diagram just shows you how they were sort of split up across the different zinc treatments. And also that there were 165 genes that were upregulated significantly in the mycorrhizal plants, regardless of zinc treatment. So this is kind of like a core set of genes that gets upregulated in a plant simply because it's colonized by mycorrhizal fungi. And then there were another roughly 400 genes that were downregulated in mycorrhizal plants. So I had this list of, nearly 1,400 genes, and I needed to narrow it down to start looking a bit more closely for genes that might have be able to answer my research question. So one of the things I did was make a list of all the ZIP genes. Sorry, I should say all the genes that were annotated as ZIPs. So not all of these have been confirmed to be ZIP transporters, but they were annotated as ZIPs. If you remember, these are the transporters that are responsible for zinc transport in the direct pathway. And then you can see from this nice heat map that these first 3 columns are the mock inoculated plants at different zinc treatments, and then the mycorrhizal plants at 3 different zinc treatments. And basically the darker the color is, the more highly expressed that gene is relative to the other treatments in this heat map. And then down the side, I've got all the ZIP transporters, genes. And so there are 2 that stand out. One you can see here was expressed in the mycorrhizal plants and not expressed at all in the mock inoculated plants. And there was another one which showed a similar expression pattern, but there was some expression in the mock inoculated plants as well. So this one, ZIP7, has been previously characterised as being able to transport manganese, and so therefore was not really of primary interest to me because there's already evidence to suggest that zinc is not its primary transport function. However, ZIP14 was really quite interesting to me. This expression profile is quite stark. We see when you look at MTPT4, which is the phosphate transporter involved in the mycorrhizal pathway, you see almost no expression in the mock plants. So this is quite similar to that. And it had an unknown function. From all the searches I did, I could find no characterization of ZIP14 yet. So I pursued this particular gene. There was also some other genes I pursued, but eventually it came down to really focusing and honing in on ZIP14 and its function. So in order to sort of get some more evidence for hypothesis 1, which is about that upregulation of expression in mycorrhizal plants, I found some other plant material that I had, and which is also metacargo plants colonized by mycorrhizal fungi or mock inoculated, grown at some different zinc concentrations, and measured the expression of ZIP14 on them, and they showed a very similar expression pattern to the RNA sequencing, which is basically not expressed at all in the mock plants and expressed quite variable, but all had some expression of ZIP14 in the colonized roots. And this is the same, well, this was the case across different zinc treatments. So I was pretty happy that I had collected enough evidence to support hypothesis 1, which was that ZIP14 was upregulated in mycorrhizal plants more than non-mycorrhizal. So then I moved on to the second hypothesis, which is about that zinc transport function. And to do this, I obtained a yeast strain, which is called ZHY3. and it has had two, ZERT1, ZERT2, are its zinc transporters. So it's had its zinc transporters knocked out. And this yeast strain cannot grow at low zinc concentrations. And then what I did was take that strain and clone ZIP14 into it to see whether that would restore the growth of yeast on low zinc media. So then I grew these yeast strains in Y&B, liquid media, and used 1 millimolar EDTA, which is a chelator. Basically, it chelated any existing zinc that was in that Y&B and allowed me to re-add zinc in different concentrations. So this work, the liquid media work, was done by Sunita, who's our co-author on the manuscript. And she did some really nice work growing the yeast, I should say, for 66 hours. And you can see this is the 0 millimolar zinc. So this is EDTA only, there's no zinc in this media, and neither of the yeast strains grew. When we added back a little bit of zinc, and this says 0.1 millimolar of zinc, but actually the amount of free zinc in there is much, much lower than that because of the EDTA. So it's actually very, very small, very, very low concentration of zinc in this one. And you can see that the mutants didn't grow over that 66 hours. However, the mutant with MT-ZIP14 did manage to grow at this very low zinc concentration. So that provided some strong evidence that there was a zinc transport function of ZIP14. And then we just measured optical density, which is a proxy for yeast growth at the end of the growing period at several different zinc, so even higher zinc concentrations. And I just wanted to show you this figure because this shows quite nicely that the complemented yeast grew. However, there's a really interesting pattern with this transporter, which is up to about 0.5 millimolar added zinc. It really improved growth over the mutant, the mutant, but then this drops off once it gets past a certain zinc concentration. So there's almost like a toxicity effect in the complemented yeast strain once it gets to higher zinc concentrations. And on the other hand, the mutants with the empty vector just kept growing even at these high zinc concentrations. So I think that's something that could potentially be examined a bit further. So, so that was hypothesis 2. We found evidence that there was the zinc transport function of ZIP14. So the next thing that I did was to try to get at where this protein is actually localized in colonized cells, to see whether it's localized where the periarbuscular membrane is, and so therefore help solidify that hypothesis as to whether that's the role it's conducting is through that mycorrhizal pathway, zinc uptake. So I cloned ZIP14 into a construct that had GFP, which is a green fluorescent protein, and then into agrobacterium. And this allowed me to do a hairy root transformation of Medicago truncatula. So basically put that construct into Medicago. You can see these are just some Medicago plants here that have just had the transformation done and are growing their hairy roots out. And then once I'd hardened those off, those plants went into soil that was inoculated with Rhizophagus irregularis, which is the mycorrhizal fungi used in all of these studies. And I grew them for around 5 weeks before washing out the roots and sectioning them and doing some confocal imaging to see whether we could see that GFP anywhere in colonized roots. So this is just an example of what we saw on the confocal microscope. So basically this is the transmitted channel, which is just, you're able to see what's there in front of you in terms of under light. So these are root cells here, this is cortical root cells, and this is an arbuscule inside a cortical root cell. You can see some intercellular hyphae on either side here as well, going into the adjoining cells. And the GFP channel shows quite nicely that our protein looks to be co-localized with where the arbuscule is. So this was really nice, and we saw this across different plants and also about 4 different transformation events. So just to ratify that what we were seeing is true, or at least we could see it across lots of different biological replicates. So there was strong evidence that ZIP14 is localized to where the periarbuscular membrane is, which is around these arbuscules here. You can also see there is no GFP in these intercellular hyphae, which is, we would not expect empty ZIP14 to be localized there, as that's fungal rather than plant material. So that kind of rounded up hypothesis 3, and now we're getting to the last one, which was looking at any potential phenotypes in plants that do not have a functional ZIP14. So to do this, I went to the TNT1 insertion mutant collection that's held by the Nobel Research Foundation or Nobel Research Institute in the US. And basically, you can go in and order lines of Metacargo truncatula that have a TNT1 insertion in your gene of interest, and that insertion should disrupt the expression of your gene, so you can essentially have knockout lines of your gene in your gene. So I ordered quite a few different lines that had insertions in ZIP14. You can see all these plants growing here, they're just waiting to be genotyped. A lot of the lines I ordered didn't come with homozygous mutants, unfortunately, so I wasn't able to use them, but 2 of them did. So with 2 lines, I back-crossed those to the wild type and then re-isolated homozygous mutants in that insertion mutation. So that just helps to clean up the genetic background of these mutants, because they may have other insertions in other genes as well, and it helps to clear up those. And then once I had those 2 lines, I measured the expression of ZIP14 in the roots just to confirm that that expression was knocked out. And what I found was actually in one of the lines, the expression was knocked down rather than knocked out. So here we've got plants colonized by mycorrhizal fungi or mock inoculated. And then the wild type, which is the segregating wild type for each line, which has a functional ZIP14, compared to the mutant that has a non-functional ZIP14. So you can see for one of my lines, the expression of ZIP14 was completely knocked out, and it was the same in the mock plants. However, in the other line, it was only knocked down, it wasn't completely knocked out. So I actually continued using both of these lines because I thought it'd be really neat to see whether, if there's a phenotype, whether it still holds true if the gene is knocked down as to whether it's knocked out. I should mention, I forgot to mention a few slides ago, but these, all of these data, they're not published yet. However, I have put the manuscript on the preprint server on bioRxiv, so you can find the manuscript and all of these figures there. I've just made a tiny URL for that. Hopefully you can see it on the screen there. So then I moved on to setting up some growth experiments to look for any plant phenotypes. So as I said, I had one knockout line, one knockdown line. I inoculated those plants with Rhizophagus irregularis or a mock inoculation, and I grew them in a low zinc soil. And so the low zinc soil was just to see whether having a functional ZIP14 conferred any advantage to those plants, which it should if they're able to take up more zinc than the plants with non-functional ZIP14. So at the conclusion of those experiments, which I repeated 3 times, measured shoot biomass, shoot zinc accumulation, and the colonization of roots by the AM fungus. So starting with shoot biomass, you can see in every panel we'll have the knockdown and the knockout next to each other, and colonized by mycorrhizal fungi, not colonized, and the wild type in blue with the mutant in pink. So the first thing you should notice here is that the wild type plants did indeed have more shoot biomass at the end of the experiments than the mutants without a functional ZIP14. And this was true both for the knockdown and the knockout plants. So it's quite a strong phenotype. And at least for the knockout genotype, the biomass was similar to that of a plant that wasn't colonized by mycorrhizal fungi at all. When we look at zinc accumulation in the shoots, there was quite a strong phenotype in the knockout genotype, with the wild type having much more zinc accumulated in the shoots than the mutant did. And it was a similar situation in the knockdown genotype, but with just not such a strong phenotype. So one thing you might have noticed, and is quite interesting, I think, is that the mutant plants colonized by mycorrhizal fungi still had more zinc than the plants that were not colonized at all, even though all of these plants basically had a non-functional ZIP14. So there was still some advantage to being a mycorrhizal plant, even without ZIP14, in terms of accumulating zinc in the plant tissues. So I have a couple of hypotheses around why that is. One of them might be that there is still another transporter on that periarbuscular membrane that has the capacity to transport zinc from fungus to plant. So therefore there's still some zinc getting through the mycorrhizal pathway, even without ZIP14. And the other theory is that it's an indirect effect of being a mycorrhizal plant. For example, if the mycorrhizal plants, the mycorrhizas are exuding things into the rhizosphere that's helping to solubilize or make zinc more available in the rhizosphere, then that may have the flow-on effect of that plant accumulating more zinc, but just not through the mycorrhizal pathway. So that's a couple of theories. So that's the plant phenotype. If I just move now to what I saw looking at the colonization of roots in these genotypes. Interestingly, I found that there was also a phenotype in terms of AM colonization. So this is vesicular colonization. So that's the proportion of vesicles, those little structures, the bubble structures I showed back in the introduction. There were significantly less vesicles in those mutant plants. So they were colonized by mycorrhizal fungi, but they had fewer vesicles than the wild type. If I look at the arbuscules, which is where that nutrient transfer is occurring, a similar situation, not quite as strong a phenotype, but still more arbuscules in the wild type than in the knockout and the knockdown genotypes. However, when we look at hypocolonization, which is really just not a functional structure per se, but just the hyphae running through the roots, there was no difference at all between these genotypes in either the knockdown or the knockout genotype. So this is really quite interesting because it suggests that knocking out ZIP14 function doesn't affect colonization per se. The plants are still able to get colonized, but it has some effect on how that symbiosis actually functions, because these functional structures, arbuscules and the vesicles, are reduced in the MT-ZIP14 mutants. So that was quite neat to see a phenotype from the fungal side as well. So just to kind of draw that to a conclusion, go back over those 4 hypotheses, I found that ZIP14 was highly expressed in mycorrhizal roots, almost not expressed at all in the non-mycorrhizal roots. When we put it into a zinc system, it transported zinc, so it was able to complement that mutant and restore growth to the yeast. And it appears to be localized, co-localized with arbusculated cells, which would suggest that it's localized to the periarbuscular membrane. And also saw some phenotypes in plants that had a non-functional ZIP14, and both from the plant side in terms of lower shoot biomass and lower total zinc in the shoots, but also from the fungal side with that reduced vesicle proportion and reduced arbuscules as well. However, the actual total colonization, if you look at the hyphae, was not disrupted at all. So I just want to conclude by acknowledging my co-authors and the people who had really a lot of input in this. in this sort of study that went for 4 years to find and characterize ZIP14. So Steffi in particular was really helpful when it came to the confocal imaging, and her expertise was invaluable there. Sunita, as I mentioned, conducted the liquid yeast work and was very helpful, giving me lots of advice on that as well. And Oliver was responsible for the RNA sequencing bioinformatics. And he made that really nice heat map that I showed you earlier. And then Matt, Jim and Steve for providing lots of support over those 4 years as part of being part of the Plant Energy Biology Centre of Excellence. And I'd really like to acknowledge the Ramsay Fellowship and the Ramsay family who supported that fellowship for 4 years. It allowed me to really do this really cool sort of blue sky research and find this gene, and also the Australasian Mycological Society, who provided the research grant that particularly funded the loss of function plant mutant studies. So thank you very much, and thanks for listening.
Thank you, Steph. That was really interesting and definitely a very well organised talk. Thank you. You stepped us through your work very well, and yeah, definitely learnt a lot. So I think if we could give you a round of applause, we would.
Thank you. I'll just imagine it.
Yeah. There we go. There's some nice emojis that are popping up. Okay, so we've got a couple of questions popping up. I might start with one though. Now, I'm not sure if I've got this on the wrong track. I'm not an expert when it comes to reading expression figures. However, why do you think there's a variation in the ZIP14 expression across the different zinc concentrations? I would have expected it to show a more linear progression. Do you think there's a reason for that? That's tricky to know. I guess the only thing that I can say about that is that when we did the radioisotope tracing to look at how much zinc was coming through the mycorrhizal pathway previously, there was also no relationship between amount of zinc in the soil and how much was coming through the mycorrhizal pathway. Like you might expect, when zinc is low, you would get more through the pathway, and when it's high, you would get less. And so the expression of ZIP14 doesn't really make sense, but it kind of matches up with what we've seen with the physiology, which is the plant doesn't seem to be able to control how much zinc is coming through, and that might somehow link back to how much ZIP14 is expressed in the roots. But yeah, it's something that I can't explain. Yeah, interesting. Well, we've got a message from Matt Solomon. Could ZIP14 be overexpressed in crops to increase zinc concentration when grown in low zinc soils?
That's a really nice question. Thanks, Matt. Yeah, I guess sort of thinking more broadly about the implications of this research, the idea is if we can sort of work out what all the different components of this nutrient transport toolbox or the pathway of uptake are, then eventually we can use that information to make crops that are more nutrient efficient, or that are able to use the mycorrhizal symbiosis more efficiently to get maximum nutrients, basically exploit the symbiosis. So from a zinc perspective, it would be really interesting to overexpress this gene and see what happens. And I think if I had more time, then I would probably try that. But there's also a lot of caveats, I think, especially with micronutrients that can be toxic as well, that if you start overexpressing them and get too much zinc in the plant, suddenly you have a negative effect of toxicity on the plant as well. Or you might just simply throw out all the other nutrients out of balance as well. So suddenly, you know, your zinc and phosphorus ratio is all out of whack as well. I would love to try it just to be out of curiosity, but I think from a crop or agriculture perspective, it'll be quite difficult to simply just overexpress ZIP14 by itself. There'd have to be other components considered as well.
Interesting. Thanks for the question, Matt. All right, from Carl Ramirez. In places where zinc deficiency is common, is it a good idea to inoculate farmland with AMF to help crops uptake more zinc? Or is zinc deficiency more due to zinc poor soils instead of low zinc uptake by crops?
It's hard to answer that generally because it could be one or the other, I guess. to know that you would have to test the soil to see what the available zinc concentrations are. And if they're reasonable and you're just not getting that zinc into the plants, then yes, maybe there's a case for sort of for inoculating the soil, or at least seeing whether there's mycorrhizal fungi already in the soil to try and increase the efficiency of that zinc uptake. But if there isn't the zinc there to begin with, like a lot of sandy soils just don't have the zinc in them, then it might be a case of needing some sort of fertilizer as well, or instead of mycorrhizal fungi. I think it's just really hard to sort of make a broad statement about that. It really depends on the soil. But I'd like to hope that in a lot of situations, being able to use mycorrhizal fungi more effectively would at least help to get some of the way there.
All right, awesome. The other question I had, I guess, also leading on to use in a real world setting is, other than soil deficiency or AMF as a component of that zinc transport, are there particular crops which would benefit from AMF that had these particular transporter proteins and therefore have an application for agriculture that would help?
Do you mean sort of crops that are more amenable to being colonized by mycorrhizal fungi, or do you mean sort of cloning a gene into other crops?
I guess a bit of both. I mean, are there crops that particularly suffer under zinc poor soils, for example?
Yeah, for sure. Things like rice, that's the only one that comes to mind right now, but there are definitely cereal crops in particular that would suffer from zinc deficiency in terms of yield, but also increasing the nutrient concentrations in the part that's eaten by humans as well. Like they might just lock up the zinc in the vegetative tissue rather than put it in the grain. And rice is also very mycorrhizal. So there are some species that get colonized better than others in terms of actual root colonization. And then there are species that are what we call positive responders, which means that they actually show a growth response more often than other species that might show no response, or even a negative response to being colonized by mycorrhizal fungi. So something like maize is a really positive responding crop. So that might be some, I would say rice and maize are potential cereals that could really benefit from working on this.
Excellent. All right, well, if there's anyone else that has any questions, feel free to jump in with your microphone and video, or send through a question on the chat. I think we're almost wrapping up now. The timing has worked quite well. While we're waiting for any last questions, I just wanted to say, first of all, a big thank you to Stephanie for jumping right in and being our first participant or first presenter for this series. We do have a schedule of presenters over the next few months, so feel free to have a look at that on our website. They may or may not take this exact format, depending on what we decide is a more streamlined process. But in the meantime, if you have any feedback, can you please send that through to me directly? I've just posted my email address on the chat group, so we'd really appreciate any feedback. And it doesn't look like we've got any more questions coming through, Steph, so thank you. Oh wait, we just got one there.
Wait, Sam, yep.
Okay, so Sam's asking, do you think the yeast growth inhibition at higher zinc concentrations is due to zinc toxicity, or could it be due to the ZIP14 protein functioning only as a high affinity transporter?
Yep, thanks for that question, Sam. Yeah, really good question. This is something that started asking towards the end of my fellowship when I was kind of running out of time, unfortunately. But I know you know Steve Timon quite well as well, and so we had a discussion about this, and he thought perhaps it could be a dual affinity transporter, in that it's can be high affinity at low concentrations of zinc and low affinity at high concentrations, or as you suggest, it could be high affinity only and unable to really control that import of zinc when it gets to the higher concentrations in the media, which is causing this toxic effect on yeast growth. But we don't know what the answer is necessarily. There were some plans to put the ZIP into Xenopus, the Xenopus system as well, which would give us a bit more information on that. But we kind of ran out of time before I left the wait. So that's kind of one of those pending questions. Thank you for asking that.
Awesome. Well, thanks everybody. Yeah, please feel free to join us next time, the last Wednesday of February. I believe we're going to have one of our current attendees presenting, Matt. So thanks everyone. Yeah, look forward to seeing you then.
Thanks, Tracy.
Thanks.
Bye.
Yeah.